Hi friends, today we are going to think about the basics of clinical trials. If you are studying Farm D, B Farm, whatever it is, if you are studying in a clinical side, we have to know what are the basics of clinical trials because nowadays we are mostly hearing about the things related to clinical trials. Let me see what are the basics of clinical trials. What do you mean by clinical study? Clinical study involves a research. So, in this research, we are using human volunteers. Human volunteers, also called participants, and we are using our medical knowledge. So, we can tell that clinical trials are a type of systematic study. In this study, we are using either a new drug or biological or devices in human subject. So thereby we are generating the data. For what is the purpose of generating data? For verifying or discovering our clinical claims. For example, if the drug is having a new anti-arrhythmic effect or it is not having adverse effect. So we are either discovering or the clinical claim such as pharmacological and adverse effect which aim to determine the safety and efficacy of a drug in the question. That is the purpose of doing clinical trials. Clinical research can be two types. First one, therapeutic research, and second one, non-therapeutic research. Therapeutic research is done to evaluate the safety and efficacy of the diagnostic, prophylactic, or therapeutic agents, devices, or combination. Or this research we are doing for the introduction of a new drug or a device. Second type, non-therapeutic research. So instead of finding the therapeutic effect, we are researching about the mechanism, processes, and risk of diseases. We are not concerning with the testing of new drug, diagnostic, or profile agents. So what is the basic of clinical trial? The clinical trial process we are conducting in accordance with good clinical practice guideline, and this GCP guideline was prescribed by International Conference of Harmonization and Declaration of Helsinki. If someone inquires, what are the phases of clinical trial? Means what are the stages of clinical trial? So five phases of clinical trials are there, but you have to know there are five stages, but the numbering start from phase 0. So phase 0, 1, 2, 3, and 4. Phase 0 means exploratory investigational new drug or microdosing study, and phase 1 human pharmacology and safety, phase 2 therapeutic exploration and dose ranging, phase 3 therapeutic confirmation or comparison, and phase 4 post marketing surveillance. So this diagram will help you to write about phase 0 to phase 4. 0, we are noting the effect on the body. In phase 1, we are carrying out for weeks, phase 2 for months, phase 3 for years, and phase 4 is an ongoing experiment or research. Whereas in phase 1, we are only using 10 participants. For phase 2, hundreds. So in phase 3 become 1000, phase 4 become number of volunteers. In phase 1, we are finding the safety in humans. In phase 2, we are checking the effectiveness at treating diseases. And in phase 3, we are checking large scale safety and effectiveness. And in phase 4, we are checking the long term safety. So, what do you mean by phase 0? As we said, phase 0, we are using or participating only less or very small number of people, normally less than 50. So phase 0, otherwise known as exploratory investigational new drug study or micro dosing study. So the use or goal of our study is to find out whether the drug will work as desired in human, means as per our expectation whether drug is working or not. In this case, in phase 0, we are using a very low doses, generally 1 by 100 of the estimated human dose or maximum of 100 microgram. So 100 microgram is the maximum dose. And we are checking the pharmacokinetic using sophisticated instrument that is known as AMS, accelerated mass spectroscopy, using the radio labeled drug, so that we can find out the ultra low drug levels. And this study normally we are carrying out maximum, or we are using normally for a time period, for example 7 days. And this phase 0 is done usually early phase of phase 1 clinical studies. And using this we can check the feasibility for the further development of the investigational new drug product. So what is the use of doing the phase 0? By doing the phase 0, we can check whether the drug pharmacokinetic and pharmacodynamic continue the exploration. For example, researchers, by using this phase 0, they can tell whether the drug enter the bloodstream or whether it is interacting, or like that we can find using phase. Next comes the phase 1. Phase 1 we are using in, or otherwise known as human pharmacology and safety study. Instead of 10, we are using 25 to 100 healthy participants we are using, and we are trying to use the volunteers or participants with particular disease, whether the new drug is good for them or causing any effect like that. Normally in phase 1 we are having many trials, especially for the anti drugs. So what we are finding is safety reporting. So if any problem is there, we have to report the safety as well as adverse reaction as per the ICS GCP guidelines. And in adverse reaction, we are checking whether it is severe, serious related. So severity like mild, moderate, severe, seriousness, whether the reaction is serious or non-serious, relatedness, possibly related, like that. We are using some terms to report safety. In phase 1 study, we can shortly tell it is done to verify the safety and tolerability of the candidate drug and to find out the maximum tolerated dose, that is MTD. So by using the phase 1 study, we can find out the safety and tolerability to determine whether animals and humans showing any significant pharmacokinetic differences, and we can also find the safe clinical dosage range. Normally we are using 1 by 5 or 1 by 10 of the maximum tolerated MTD dose in animals as per the. In phase 1, we are trying to determine the pharmacokinetics of the drugs in human, whether to detect any predictable toxicity is there. Normally, this is carried out for a duration of 6 to 9 months. And phase 1 study we are usually conducting using non-blind or open label. Approximately, if you are doing the drug, 70% of the drug will move from phase 1 to phase 2. So, what is phase 2? This phase 2 is also known as therapeutic exploration and dose ranging. In this phase, the drug is tested for the first time for the patient with a target disease, and we are finding out the therapeutic efficacy, dose range, and ceiling effect. So we can tell that phase 2 is the extension of phase 1. In phase 2, we are finding out some endpoints like surrogative, definitely some words. Surrogating means any predictive endpoint is there. If the if there is any reduction in tumor cells, for example in case of cancer cell, definitely means we can measure the direct size or how much pain is relieved, like that. Such endpoints can be done with the help of this phase 2 trials. In phase 2, several hundreds of participants are participating in the trial, and we can find out the therapy safety, side effect, potential risk. So what we are doing is that patients are carefully monitored and assessed continuously in phase 2. In this phase, we are checking for the tablet, extended release capsule, infusion, etc. And phase 2 trials are discontinued if the drug is not showing any effectiveness or have any safety problems that we are not moving. The duration of the phase 2 is 6 months to 3 years, and this phase also divided into phase and late phase. In early phase 2, we are using or we are carrying out in approximately 200 participants, and we are checking the dose range. Usually single blind is using, or sometimes we are using randomized double blind also using find the effect. But in the second phase, short phase 2 trials, we are doing in carefully conducted blind studies in a same type, that is homogeneous population, to ensure our safety and efficacy. From the phase 2, approximate 33% only moving to the phase, that is phase 3. Phase 3, otherwise known as therapeutic confirmation or comparison. Here, instead of hundreds, several hundred to thousand patients are involving in phase 3. So the population is increasing, and we are carrying out the randomized double blind study here. Here what is using whether the value of the drug is useful for existing therapy. We are confirming. So by this trial, we are able to minimize the errors in the information what we have gathered in phase 1 and phase 2. Phase 3 trial with the crossover design. So this is the crossover design. So we are categorizing the patient group into randomized into 3 group, and we are changing randomized. If we are giving in the week 1 standard, so we are giving in the changing the order and the medication. In phase 3, it is carrying out for the years 1 to 4 years. Out of this, 25 to 30 percentage population will move to the next phase. Again, the phase 3 is subdivided into 3A and 3B. In 3A phase studies, we are carrying out for the approval of the drug from the appropriate regulatory agency. For example, if you are doing NDA application or NDA application, we are carrying out the phase 3A. Most of the phase 3 studies are conducted in phase 3A trial. But in phase 3B study, we are carrying out or we are starting before the marketing. And phase 3B studies will give an additional safety data before going to the market. So we can tell that this phase 3 studies are additional studies which will help to gather additional information. And we are using the volunteers as a large heterogeneous patient population to produce the maximum data. Then comes the phase 4, that is known as post-marketing surveillance. This study begins after getting the marketing approval, and this post-marketing surveillance is a practice of monitoring the drug safety or medical device after when we release the drug into the market. So we are taking a broader population for the effect. So what we are finding out is or monitoring the long-term safety and efficacy, and we are confirming the drug or the device good effect. So phase 4 trial include additional drug-drug interaction, dose response, safety trials, all will come under this PMS. Usually uncontrolled observational studies are done in phase 4, and the duration of the study depends on the purpose of the study, and it may be indefinite. It will be extending for a number of years. So by this, I think you will get an somewhat idea about the clinical trials and various phases and what are the uses of doing the clinical trials. Hope this will be helpful for you. If you find useful, please share to your friends. Thank you for watching.